Psychedelic therapies are getting a lot of attention these days, and for good reason.
Yep.
They have huge potential,
Yeah.
to change how we treat mental health.
Yeah.
But before all that can happen, before we can unlock that potential, we need to make sure they're safe for patients.
Yeah.
And that's where things get a little less exciting. Maybe not as glamorous, but it's super important.
Right.
We're talking about impurity assessments. Impurity assessments in how we develop new drugs.
Yeah, it's a critical part of the process.
So today we're diving deep into the world of, well,
impurities.
Impurities, yeah.
Yeah.
Especially these two types, nitrosamine and genotoxic impurities. And we're going to focus on why these assessments, these checks for impurities are so important, not just to keep patients safe, but also to navigate those regulatory hurdles.
Yeah, getting those new drugs to market.
To market, exactly. And that's especially true for these new psychedelic therapies.
Right, right. And what makes this whole topic so interesting, is that it's this kind of meeting point where cutting-edge science meets these really strict safety protocols. Right, you're talking about drugs that could really change lives, but
yeah,
we have to be so careful. Meticulous analysis every step of the way.
Absolutely. So let's start with these nitrosamines.
Okay.
They have, I guess you could say, a bit of a reputation, not a good one.
Right.
The unwanted guests at the party. Why are they causing so much concern?
Well, some nitrosamines you see are what we call carcinogenic.
Okay. So that means
they can cause cancer.
Oh, okay. Got it.
And what makes them tricky is they can pop up unexpectedly.
Unexpectedly?
Yeah, during drug manufacturing.
Oh, I see.
And it's often a byproduct.
Byproduct?
Yeah, of reactions involving anomines.
Anomines, huh?
And those are, common building blocks.
Building blocks.
In a lot of pharmaceuticals.
Ah, so it's not just like contamination, like from the outside.
Not always, no.
These nitrosamines can just appear during the process itself.
Exactly.
Wow. And that's why they're so hard to control.
It is. It's like, imagine you're baking a cake. You carefully chose your ingredients. You followed the recipe perfectly. Right. But then as it's baking, this weird taste develops. You didn't put it there, but there it is, affecting the whole cake. That's nitrosamines. They can just show up, affecting the final drug.
Oh, that's a great way to put it. It really does show how complex this all is.
It is complex, yeah.
So now let's shift gears a little. Talk about these genotoxic impurities.
Okay.
These sound even scarier. What makes these substances so bad?
Well, genotoxic impurities can damage DNA, the genetic blueprint of our cells.
Right.
And even tiny amounts.
Even tiny amounts.
Yeah, it can be risky. Because when DNA gets damaged, it can lead to mutations. Oh. And sometimes those mutations lead to cancer.
That's pretty scary.
Yeah. Think of DNA like this really important instruction manual for our bodies.
Okay.
Even a little rip or a smudge on a page can mess things up.
Wow. That's a good analogy. Our bodies are pretty vulnerable then, to these tiny invisible threats.
We are, yeah.
So given how serious these risks are, I'm wondering about the rules, the regulations. What are the expectations for managing these impurities?
Good question. Regulatory agencies like the FDA and the ICH, that's the International Council for Harmonization.
Oh, right, the ICH.
They have very strict guidelines to mitigate those genotoxic risks.
Makes sense.
A key document is ICH M7.
ICH M7.
It's all about controlling DNA reactive impurities in pharmaceuticals.
Okay.
So it provides a framework.
A framework.
Yeah, for assessing and controlling these impurities.
Okay. And this framework, it uses a risk-based approach, right? Meaning it focuses on the impurities that pose the biggest threat to patient safety, not every single impurity.
Exactly. You got it. It's not about getting rid of every single impurity. That would be impossible. It's super expensive. It's about figuring out which ones are most likely to cause harm and focusing on those.
Okay, so how does that work? Like, how do they decide which impurities are the riskiest?
Well, they look at a few things. The impurity's chemical structure, whether it can interact with DNA, and the levels we might find in the final drug.
So it's about prioritizing those impurities that are most likely to cause harm and then tailoring our control strategies accordingly.
Exactly. It's all about using our resources effectively and focusing on what matters most. And that's where those sensitive analytical techniques we discussed come in.
Right.
ICH M7 actually provides guidance on how to set appropriate limits for these impurities, what we call acceptable intake limits. based on how toxic they are and how much a patient might be exposed to.
Okay, so it's not just about finding these impurities, it's also about measuring them and seeing if those levels are safe.
Precisely. And this information helps guide the development of those control strategies we talked about, preventing formation, testing, optimizing the process, all to keep those impurities below the acceptable limits.
And I remember you mentioned a book earlier, Mutagenic Impurities. How does that fit into all of this? Does it help with applying those ICH-M7 guidelines?
Absolutely. It's like a practical guide.
A practical guide, okay.
To help you use those ICH-M7 principles in real-life drug development situations.
Right.
It was written by Andrew Teasdale, a real expert in this field. It breaks down how to apply those guidelines.
Okay, so it kind of translates those complex guidelines into something actionable.
Exactly.
So anyone working on this is their go-to resource.
It is. It gives you strategies for identification, for controlling those impurities, and for assessing the risk.
That sounds really helpful.
It is. And as we dive deeper into the nitrosamine and genotoxic impurity assessments, we'll see how Teasdale's insights and what's in ICH-M7 come into play, especially when we talk about proactive risk assessment and how to reduce those risks.
Okay, so we've laid some groundwork here. We understand why these impurities are a problem. And we have an idea of the regulations involved. Now, let's get specific.
Okay. Yeah.
Let's start with nitrosamines. How do we find them in drugs? And what can we do to keep them in check?
Well, detecting nitrosamines is tough. It takes really sensitive techniques.
Sensitive, huh?
It's like finding a tiny grain of sand on a huge beach.
Wow.
You need the right tools to even see it. So we use chromatography.
Chromatography.
It's a way to separate and analyze different parts of a mixture.
Okay.
And we combine that with really advanced detection methods, like mass spectrometry. And then we can pinpoint and measure even the tiniest amounts of nitrosamines, whether in a drug substance or the final product.
So it's like having a super powerful microscope that can zoom in and spot those hidden impurities.
That's a great way to think about it, yes. And once we know nitrosamines are there, we can use strategies to control them.
Control them.
Yeah. And these strategies usually fall into 3 categories.
3.
Preventing their formation, doing analytical tests, and optimizing the process.
Okay, let's break that down a bit. Starting with preventing formation. What does that involve?
It's all about minimizing the chances of nitrosamines even forming.
I see.
So that means choosing the right starting materials and reagents. Avoiding the ones we know can easily form nitrosamines.
Okay.
And then we have to carefully control the reaction conditions. Things like temperature, pH, and even the presence of certain catalysts. All of these can affect whether these impurities show up during manufacturing.
So it's about being proactive and designing the process from the beginning in a way that keeps those impurities out.
Exactly. You want to make it really hard for those unwanted guests to show up.
Right.
And this is where a deep understanding of chemistry is so important. Scientists need to think about how reactive different chemicals are.
I see.
And how they might interact and, maybe accidentally form nitrosamines.
Okay, so preventing formation is like step 1. What about those analytical tests you mentioned?
Oh, they're crucial. We use those tests to double-check that our prevention strategies are working.
Makes sense.
And to catch any tiny amounts of nitrosamines that might still slip through.
Okay, so it's like a quality control checkpoint.
Yeah.
Throughout the manufacturing process.
Exactly. We don't just test at the end. We test at different stages.
Ah, so it's ongoing.
It is, from the raw materials to the intermediates to the final drug product.
Yeah.
And we use those sensitive techniques like chromatography and mass spectrometry.
Right. To track the nitrosamine levels throughout the whole process and hopefully catch any potential problems early on.
Exactly. It's like a system of early warning signals.
I like that analogy.
Yeah, it helps us catch those problems before they get big.
Okay, so we've talked about preventing formation and testing. Now, what about process optimization? How does that help with nitrosamine control?
So process optimization is all about refining the manufacturing process itself.
Refining it.
Yeah, making it even better so that it reduces the chances of nitrosamines forming.
I see.
This might involve adjusting reaction conditions.
Okay.
Maybe exploring different ways to synthesize the drug.
So finding a different route, so to speak.
Exactly. Or even just changing the order of steps in the process.
So it's all about fine-tuning those little details, making it as hard as possible for those nitrosamines to appear. That's fascinating.
It is. And it often takes a team effort. You have chemists, engineers, analytical specialists all working together.
Right. They
They identify areas where the process can be improved, and then they implement those changes. It's pretty amazing.
It's a real team effort to outsmart those pesky nitrosamines. Now, I'm curious about how all this applies to psychedelic therapeutics, the ones we talked about at the beginning,
Right.
they seem to be particularly susceptible to nitrosamine formation. Why is that?
You're right. They are. And that's because many of them contain secondary amines. It's part of their chemical structure. And those secondary amines, they're very reactive. They can easily react with other things during synthesis.
Oh,
And when they do, guess what forms?
nitrosamines.
You got it.
So it's like a built-in vulnerability,
You could say that.
making them really challenging to work with.
It is a challenge, yeah. But it's also what makes this field so exciting. We're pushing the boundaries of what's possible, exploring totally new ways to treat people.
Right. It's like venturing into the unknown.
Exactly.
And that comes with its own set of challenges.
Right.
But also amazing potential rewards.
I agree.
Now, before we move on to genotoxic impurities, I want to touch on one more thing. Proactive risk assessment. How does that fit into this whole nitrosamine control strategy?
Proactive risk assessment. That's the cornerstone of drug safety.
It is.
Absolutely. It means identifying where those nitrosamines might come from,
Okay.
understanding how they form,
Right.
and figuring out how likely they are to end up in the final drug.
I see.
It's like being a detective.
A detective.
Yeah. You investigate everything before you even start working in the lab.
So you anticipate the problems.
Exactly. And then you come up with strategies to stop those problems before they happen.
So it's a shift from reacting to problems to preventing them in the first place.
Exactly. And that's where ICHM-7 comes in.
Right. Guidelines.
It gives us a framework for doing those risk assessments thoroughly. And Teasdale's book, Mutagenic Impurities, it really complements those guidelines, gives practical advice, real-world examples.
So if you're working on this, those two resources are essential.
They are.
Okay, so we've covered a lot about nitrosamines, how they form, how we detect them, and all the strategies for keeping them under control.
We have. It's a lot to take in.
It is, but it's fascinating stuff. Now let's move on to those genotoxic impurities. What are the specific challenges there?
Yeah.
And how are the control strategies different?
Right. Well, as we talked about before, genotoxic impurities can damage DNA.
Right. A big risk to patient safety.
Exactly. And while there are some similarities with nitrosamines in how we control them, there are also some key differences.
Okay, I'm curious to hear about those differences. What makes genotoxic impurities especially tricky to manage?
One of the biggest challenges is their potency.
Potency.
Meaning even tiny amounts can damage DNA.
Wow.
So our control strategies have to be incredibly sensitive and effective.
Oh, I see. So those microscopic lenses we talked about before need to be even stronger for these impurities.
Exactly. We have to be able to find even the tiniest traces of them to make sure our controls are working. And another challenge is that there are so many different kinds of genotoxic impurities.
Oh, so unlike nitrosamines, which are all part of the same group, these impurities are more diverse.
You got it. They can come from all sorts of sources, and they have different chemical structures.
So that makes it harder to develop one strategy that works for all of them.
It does. We have to tailor our approach for each specific impurity. It's more individualized.
It sounds like managing genotoxic impurities requires a really deep understanding of both chemistry and toxicology, the study of poisons.
It does. We need to understand how these impurities react, how they might form.
Okay.
But we also need to understand how they interact with DNA,
Right.
what kind of damage they can cause. That knowledge is key to developing the right control strategies.
Okay, so we've established that genotoxic impurities are really potent, and they come in many forms. And that means we need highly sensitive detection methods and specific control approaches for each one. Now let's dive into those control strategies. What are some of the key approaches for reducing the risks associated with genotoxic impurities?
Well, just like with nitrosamines, the main goal is to minimize their presence in the final drug product.
Right, of course.
We use a combination of strategies, similar to the ones we talked about for nitrosamines, but with some adjustments.
Okay.
So let's start with formation prevention. How does that work with genotoxic impurities?
Okay, formation prevention, that sounds familiar.
It's the same idea. We want to prevent these impurities from forming in the first place. So we're careful about which starting materials and reagents we use.
Right.
We avoid anything known to be genotoxics.
Makes sense.
Or anything that might create genotoxic byproducts during manufacturing.
So it's about choosing your ingredients wisely.
Yeah.
Making sure they have a clean record, so to speak.
That's a good way to put it, yes. And it also involves meticulously controlling those reaction conditions, things like temperature, pH, and catalysts. They can all affect whether those unwanted side reactions happen.
Right, the ones that might lead to genotoxic impurities forming.
Exactly. It's all about setting the stage for a clean and controlled process from the very beginning.
So it's about being proactive and
Mhm.
setting up the process in a way that makes it really difficult for those impurities to even form. What about analytical testing? How does that fit in with managing these impurities?
Analytical testing is absolutely vital.
Vital.
It is. We need to make sure our prevention strategies are actually working.
Right.
And we also need to detect any genotoxic impurities that might sneak in, even if we've done our best to prevent them.
I see.
And remember, these impurities are super potent.
Right.
So even small amounts can be a problem. That means our analytical techniques have to be incredibly sensitive.
So even more sensitive than for nitrosamines.
Yes, exactly. It's like those microscopic lenses we talked about, but with even higher magnification. We need to be able to see those tiny, tiny traces of impurities to guarantee the final drug is safe.
And what are some of the techniques used to do that?
Well, there are a few, but some common ones are high-performance liquid chromatography, or HPLC,
I'm looking PLC.
and gas chromatography, or GC. And we often combine them with mass spectrometry, or MS, to make them even more sensitive and accurate.
So these techniques help identify and measure those genotoxic impurities, even if they're only present in tiny amounts.
That's right. And we use these tests at different stages of the manufacturing process, too.
Oh, right. Just like with nitrosamines.
Yeah.
On the raw materials, the intermediates, and the final drug product.
Exactly. That way we can track the impurities and make sure the levels stay within those acceptable limits.
Okay, so I've got formation prevention and analytical testing. That's 2 down from. What about process optimization? What's its role in all of this?
Process optimization, that's where we fine-tune the manufacturing process itself. Yeah, to further minimize the chances of those genotoxic impurities forming.
Okay.
So we might adjust the reaction conditions, maybe lower the temperature or change the pH.
Right.
To make it harder for those impurities to form and easier for the drug to be made properly. We might even try completely different ways to synthesize the drug substance.
Wow.
Yeah. Sometimes there's a completely different path that bypasses those tricky steps where impurities are more likely to form.
So it's like finding a detour around those hazardous areas.
That's a great analogy.
And all of this requires a real team effort, right?
It does. You have chemists, engineers, analytical specialists all coming together to brainstorm, experiment, and implement those changes.
So it's really a collaborative effort to outsmart those impurities.
It is, and it requires a deep understanding of the science behind it all.
Right.
And that's where Teasdale's book and the ICH-M7 guidelines are so important.
They provide that foundation, that knowledge base.
Exactly. They help us navigate these complex challenges and develop those really robust control strategies.
Okay, so we've explored this multifaceted world of controlling genotoxic impurities, focusing on preventing formation, testing, and optimizing the process.
Right.
Now, I want to loop back to those psychedelic therapeutics we started with. We know they are particularly vulnerable to nitrosamine formation, but what about genotoxic impurities? Do those same concerns apply?
That's a great question, and it highlights a really important point. While a lot of the focus on impurities in psychedelic therapeutics has been on nitrosamines, we can't forget about genotoxic impurities either.
So we need the same level of vigilance and control for both types of impurities.
Absolutely. And it's especially important because a lot of these psychedelic therapeutics are made synthetically.
Oh, right. Not naturally occurring.
Right. And that means we have more control over the manufacturing process.
Okay.
And that allows us to use those proactive risk assessment and mitigation strategies we talked about earlier.
So it's about applying those same principles.
Exactly. Carefully selecting materials, meticulously controlling the reaction conditions, and rigorously testing everything throughout the entire process, whether it's for nitrosamines or genotoxic impurities.
Okay, so we're applying those same principles,
Yeah.
but for both types of impurities.
Exactly. And it's also about staying informed about the latest research and regulatory guidance. The field of psychedelic therapeutics is moving fast.
Right.
Our understanding of these impurities is always evolving.
It's a reminder that science is constantly changing,
Yes.
and we need to keep learning and adapting to make sure patients are safe.
I agree.
Okay, so we've gone pretty deep into the world of impurities in pharmaceuticals. We've talked about the risks, the regulations, and the strategies for detecting and managing them.
We have. It's a lot, but it's all crucial for patient safety.
It is. And we've established that all these principles apply to those new psychedelic therapies that hold so much promise.
Right, those therapies that could really change how we treat mental health.
Exactly. Now, before we move on to the next stage of our exploration and talk about the specific challenges and opportunities in psychedelic therapy development, let's take a moment to gather our thoughts. We'll be right back.
So welcome back to our deep dive into impurity assessments. Before we jump into the specific challenges and opportunities with psychedelic therapies, I think it might be good to go back to ICH-M7 for a bit.
Okay.
You mentioned it provides a framework for managing these impurities. Can you explain a little more about what makes this guidance document so important for the pharmaceutical industry?
Yeah, definitely. ICH-M7 is like the roadmap for navigating the whole world of genotoxic impurity assessment and control.
Right.
It's a globally harmonized guideline.
Okay.
Meaning regulatory agencies in tons of countries use it. The US, Europe, Japan.
Ah, I see.
So it streamlines things for companies making drugs in those regions.
Makes sense.
And what's really cool is it doesn't give strict rules. It outlines a more flexible approach based on science for how we assess and reduce risk.
That risk-based approach is a really key part of ICH-M7.
Right.
Can you explain how that works in practice?
It's all about prioritizing.
Prioritizing.
Yeah, we focus our energy where it matters most.
Okay.
Instead of trying to get rid of every single impurity.
Right.
Which would be practically impossible and super expensive.
Hmm.
ICH-M7 helps us figure out which impurities are the riskiest, the ones most likely to cause harm to patients.
I see.
And we focus on those. So we look at things like the impurity's chemical structure,
Okay.
its potential to interact with DNA,
Right.
and the levels it might be present at in the final drug product.
So it's about identifying the most dangerous impurities and then tailoring our control strategies to those.
Exactly. And that's where those sensitive analytical techniques come in, the ones we talked about earlier.
Right.
ICH-M7 actually gives guidance on how to set those limits.
Limits.
Yeah, for how much of each impurity is allowed. We call them acceptable intake limits.
Ah.
And these limits are based on, well, how toxic the impurity is and how much a patient might be exposed to.
So it's not just about finding those impurities, it's about measuring them and making sure the levels are safe.
Exactly. And all of that information then informs those control strategies, the formation prevention, the testing, the process optimization,
Right.
all aimed at keeping those impurity levels in check, below those acceptable limits.
Right.
And what's particularly interesting about ICH-M7, I think, is the emphasis on understanding how these impurities form.
That's interesting. Can you say more about that? Why is understanding those formation mechanisms so important?
Because if we can understand how and why those impurities show up, we can create better strategies to prevent them right from the start.
I see.
For example, let's say we know a certain reaction condition, like high temperature or a specific catalyst, tends to create a particular genotoxic impurity.
Okay.
Well, then we can adjust the process.
Adjust.
Yeah. Avoid those conditions. Or maybe even find a completely different way to make the drug, one that bypasses those risky steps.
That makes sense.
So it's all about getting to the root of the problem.
It is. It's a very proactive approach, anticipating and preventing problems before they even happen. That's a big part of ICH-M7. And it shows how the pharmaceutical industry's thinking has changed.
Yeah, it's a real shift. And that shift is partly because of those, high-profile cases where impurities cause drugs to be recalled.
Right.
Or even harm patients.
Right.
The industry has learned a lot from those experiences.
Yeah.
And now this proactive risk management approach is really embraced.
It is.
And that brings us to another important thing about ICH-M7,
What's that?
the focus on documentation and transparency.
Ah, okay.
Can you talk a little bit about why those are so important?
Yeah, for sure.
Well, ICH-M7 is really clear. Everything needs to be carefully documented. Every step of the way.
Every step.
Every single step.
Mm.
From the initial risk assessment to choosing the control strategies to validating those analytical methods.
Okay.
And then the ongoing monitoring of those impurity levels.
Got it.
Everything needs to be clearly documented.
Yeah.
Thoroughly documented.
So it's like creating a paper trail that shows everyone you've done your due diligence. You've taken all the necessary steps to ensure safety and quality.
Exactly. And it's important for several reasons. It proves that the company has carefully looked at the risks and taken the right steps to mitigate them.
Right.
It lets regulators review the process and make sure the company is following the guidelines.
Okay.
And it makes sure everything is consistent, that future batches of the drug are made to the same high standards.
So it's all about transparency and accountability.
Exactly. And that transparency helps build trust in the pharmaceutical industry. And it helps patients feel confident about the safety and quality of their medications.
Right. It all goes back to that core principle we talked about.
Yeah.
Putting patient safety first.
Look at this.
Now, I want to go back to something you mentioned before. Andrew Teasdale's book, Mutagenic Impurities.
Okay.
How does this book work together with the guidance from ICH-M7?
Teasdale's book, it's like the perfect companion to ICH-M7.
Okay.
It gives really practical advice
Okay.
and real-world examples
Mm-hmm.
to help you understand and use those guidelines.
So it helps bridge that gap between the theory and the actual practice.
Exactly. It's like having an expert guide you through the whole process,
Right.
giving you insights and tips, and even sharing stories of what not to do based on real experiences.
I like that. So it's a really valuable resource.
It is, and I find it particularly helpful with proactive risk assessment. It provides a step-by-step approach.
Step-by-step approach.
Yeah, for identifying potential sources of those mutagenic impurities,
Okay.
understanding how they form,
Right.
and then figuring out how likely they are to end up in the final drug product.
So both ICH-M7 and Teasdale's book emphasize this proactive mindset, anticipating and dealing with potential problems before they even come up.
Exactly. And it highlights a fundamental shift in how the pharmaceutical industry thinks about drug safety.
Yeah.
It's not just about reacting to problems anymore. It's about preventing those problems from happening in the first place.
And that's especially important as we explore new frontiers in drug development, like with those psychedelic therapies we've been discussing.
Absolutely. These therapies, they have huge potential, but they also come with unique challenges when it comes to controlling impurities. And we already talked about how many psychedelic compounds contain those secondary amines.
Right, the ones that make them vulnerable to nitrosamines.
Exactly.
And we need to be just as careful about genotoxic impurities, too.
Right. So we need to apply all those same principles.
Hmm.
Careful material selection, meticulous control of reaction conditions, and rigorous testing.
Yes. Regardless of which type of impurity we're dealing with.
And it's important to remember that this is an ongoing process.
It is. It's a journey, not a destination. The field of psychedelic therapeutics is growing so fast.
Yeah.
Our understanding of these impurities and how to manage them is constantly evolving.
Exactly. And that means we need to keep learning, keep adapting,
Yeah.
if we want to ensure the safety of these new therapies.
Right.
Okay, so we've really explored the world of genotoxic impurity control,
We have.
highlighting the importance of formation prevention, analytical testing, and process optimization.
Right. All crucial aspects.
Now let's finally shift gears. Let's dive into those specific challenges and opportunities, the ones presented by psychedelic therapeutics. You mentioned earlier how many of these compounds have those secondary amines, which make them prone to forming nitrosamines.
Yeah.
So what strategies are being explored to tackle that problem?
Well, researchers are trying a bunch of different things,
Okay.
all with the goal of minimizing nitrosamine formation. During the synthesis of these compounds,
Okay.
one strategy involves actually tweaking the chemical structure of those secondary amines.
Tweaking it. Yeah. Making them less reactive. So they're less likely to form nitrosamines.
So it's like reinforcing a weak spot in the molecule.
Exactly. Another approach is to optimize those reaction conditions.
Okay.
Maybe using different solvents, changing the temperature, or even finding different ways to synthesize the drug.
Oh, right. Bypassing those steps where nitrosamines are likely to form.
Exactly. It's like finding a safer route.
Right.
And of course, we still use those sensitive analytical techniques, the ones we've been talking about, to keep a close eye on those nitrosamine levels,
Okay.
to make sure our control strategies are working.
Yeah.
And that the final drug is safe.
So it's a multi-pronged approach,
It is.
combining a deep understanding of chemistry with this commitment to, constantly improving and adapting.
Absolutely.
Now, I'm curious about the bigger picture. What impact is this intense focus on impurity control having on the field of psychedelic therapeutics as a whole?
It's having a huge impact. It's changing how these therapies are developed, manufactured, and even regulated.
Oh.
This focus on safety, it's raising the bar for everyone, the researchers, the chemists, the manufacturers, the regulators, and that's a good thing. It means these therapies, which have such incredible potential, are being brought to market with patient safety as a top priority.
So it's about finding that balance, right?
Yeah.
Between encouraging innovation and making sure safety standards are high.
Exactly. And that balance is essential for the long-term success of this whole field.
I agree.
If we embrace these challenges and work hard to find solutions, we can make sure that these groundbreaking therapies reach their full potential, while also keeping patients safe.
It's really exciting to see this cutting-edge science and these rigorous safety protocols coming together like this.
It is.
It shows how dedicated and innovative people are in this field, working hard to bring these therapies to the people who need them.
Absolutely. And as we move forward, it's important to keep talking about this, sharing knowledge, working together across different fields, and never losing sight of that core value.
Patient safety.
Patient safety. Exactly. And we're back. For the final part of our deep dive, we've been exploring this really complex world of impurity assessments in pharmaceuticals, with a special focus on those exciting psychedelic therapies.
Yeah, it's been quite a journey, hasn't it?
It has.
We started by understanding why these impurities are such a big deal.
Right. Both nitrosamines and genotoxic substances.
And then we looked at all those incredibly detailed strategies they use.
Yeah, to find them, to measure them, and to control them.
It's amazing how much work goes into it.
It is, but it really all comes down to one thing.
What's that?
Patient safety. That's the core value here.
Absolutely. All of this scientific progress, all this innovation, it has to be balanced with responsibility. We have to protect the people who will benefit from these discoveries.
Right. Their health and well-being comes first. We've talked about those strict regulatory guidelines like ICH-M7.
Right. It provides that framework for assessing and mitigating risk.
Exactly.
Yeah.
But then we also saw how those guidelines are actually put into practice with resources like Andrew Teasdale's book, Mutagenic Impurities.
That book is, it's a real gem.
It is.
So much practical knowledge in there.
Yeah, like having an expert guide you through it all.
Exactly. Sharing all those little tips and tricks.
Yeah, things you only learn from years of experience.
Right. And it emphasizes that proactive approach.
Oh, yeah, the proactive approach.
It's so central to how drugs are developed now.
Absolutely. Anticipate problems.
Right.
And prevent them from happening.
Exactly. Instead of waiting for something to go wrong and then reacting.
Right. It's about preventing those problems in the first place.
I like that. Being proactive.
Okay, so as we wrap up this deep dive, I want to bring it back to those psychedelic therapies.
Okay.
The ones that started this whole conversation.
Yeah, they hold so much potential for changing how we treat mental health.
They do. But they also have unique challenges.
Right. We talked about how many of these compounds, they have those secondary amines.
Yeah. And that makes them particularly susceptible to nitrosamine formation.
It does. But we can't forget about the possibility of genotoxic impurities, too.
Right.
So we need to apply all those same principles: meticulous control, rigorous testing, and proactive risk assessment.
Yes, all of them.
Whether we're talking about nitrosamines or genotoxic impurities.
Exactly. And we always have to remember, this field is moving so fast.
It is.
Our understanding of these impurities, it's constantly evolving.
Yes, science never sits still.
It doesn't.
So we always have to be learning, adapting.
If we want to make sure these new therapies are safe and effective.
Absolutely. It's an ongoing process.
It is.
And that's where those resources we discussed, Teasdale's book, the ICH guidelines, the latest research.
Right.
They're all so important.
They are.
They give us the tools and the knowledge to navigate these complex issues.
And to make decisions that put patient well-being first. Exactly. As we wrap up, I hope our listeners have a better understanding now of this intricate world of impurity assessments and the crucial role it plays in ensuring these life-changing therapies are safe and effective.
I hope so, too. It's a fascinating topic, even if it's a little behind the scenes.
It is.
And we encourage everyone listening to learn more, dive into Teasdale's book, check out those ICH guidelines, stay curious about the latest discoveries in this field.
And remember, knowledge is power. The more you know about the science behind these therapies, the better you can advocate for yourself for safe and effective treatments.
That's right.
Thank you for joining us on this deep dive. It's been a pleasure exploring this world with you.
It has.
And until next time, keep asking questions, keep learning, and keep exploring the amazing world of science.